This is the depolarization of a neuron, which sends an action potential down the axon to the synaptic end bulb and eventually creates a stimulus in the next cell in the pathway. So you can see that we have a larger concentration of these blue sodium ions outside the cell, a larger concentration of these red potassium ions inside the cell. And the first step in the process is a ligand from another cell binds to its receptor, which in this case is the sodium channel. It opens the sodium channel and allows sodium to flow into the cell. The resting potential of this neuron is -70 millivolts, and once the sodium travels into the cell down its electrochemical gradient, it's going to change that voltage. The sodium is flowing from a higher concentration to a lower concentration. That's down the chemical gradient, and it's also attracted to these large negatively charged proteins. So it's flowing down its electrical gradient as well, and the positive sodium ion is being attracted to the negative protein. Once the sodium flows in, it's going to raise the voltage of the cell body from -70 millivolts up to -60 or -55 millivolts. Once that voltage reaches the axon hillock here, the first voltage-gated sodium channel opens here in the axon hillock, and sodium starts to rush into the cell. This -60 or -55 millivolts is the threshold. Once that's reached, then an action potential can be propagated down the axon. The sodium rushing in causes the next area to reach threshold as well, which causes the voltage-gated channel to open, and more sodium to rush in. This propagates down further, and the voltage increases to -55 over here. This channel opens, more sodium rushes in, and this wave, or like she said, a domino effect, travels all the way down the axon. And this is your action potential. And like she said, it was an all or none principle. If -60 or -55 millivolts, the threshold level, hasn't been reached, then this action potential can't propagate. Once the action potential propagates down, enough sodium has come into the cell to raise the voltage all the way up to +30 millivolts. And once this 30 millivolts hits the synaptic end bulb, these voltage-gated calcium channels open, and calcium is allowed to rush into the cell. It is also moving down its chemical gradient, since there is a higher concentration of calcium outside the cell than there is inside the cell. The calcium rushing in triggers the exocytosis of these synaptic end vesicles, which bind to the plasma membrane, and that in turn releases the neurotransmitter or ligand into the synaptic cleft. And we remember from muscle action potentials that this in turn is going to bind onto a ligand receptor on the motor end plate and trigger another action potential, and so on and so forth. Once this pathway is over, we also have repolarization happening. And during repolarization, which starts at +30 millivolts, your voltage-gated potassium channels open, these red ones here, and they're going to open all along the cell. And they're going to allow the potassium to rush out of the cell, since there's a higher concentration inside and a lower concentration in the extracellular fluid. The potassium is going down its concentration gradient. Also, at +30 millivolts, the voltage-gated sodium channels close, so no more sodium can rush into the cell. At the same time, this ligand all the way over here is detaching from the ligand-gated sodium channels, so they're going to close as well. And then because the potassium has rushed out of the cell, we're bringing the voltage from positive 30 millivolts back down to negative 70 millivolts, and that's repolarization. And if it goes any lower, it's going into hyperpolarization. And the reason the potential is changing is because you have positive ions rushing out. And when you lose a positive charge, things become negative. So now that there's all this sodium inside the cell and all this potassium outside the cell where it shouldn't be, your sodium-potassium ATPase pumps kick on, and they're working in hyperdrive. And they're using 1 ATP to bring out, let's see if I can get my colors right here, bring out 3 sodium and bring in 2 potassium. 2 potassium and 3 sodium for 1 ATP. And those pumps are working in hyperdrive to get the concentration gradients back to their resting state, with a higher concentration of potassium inside and a higher concentration of sodium outside the cell. You also have calcium pumps down here at the other end that have also kicked on, and they're also moving calcium against its concentration gradient to move all of this extra calcium back out of the cell, so that there's a concentration gradient with a higher concentration of calcium outside the cell. The other thing that she mentioned in class was that there's chlorine, and if a certain ligand binds over here, then it will open, well, not on the sodium channels, but on the chlorine channels, on ligand-gated chlorine channels, it will open those and chlorine can rush in, and that will hyperpolarize your cell instead of depolarizing it. And then the other thing she said was the way the ligands are broken down is they can be broken down by an enzyme. They can diffuse away from the synaptic cleft, or they can reuptake can happen, and your ligand will go back into the presynaptic cell. So that is the pathway of depolarization in a neuron.